كلكم موجودين؟ Okay, ladies and gentlemen, welcome back again. And we are very happy to have Professor Volker Ehammen with us this morning. And Professor Volker is the head of flow cytometry core facility in Heidelberg University in Germany. And he was a visiting professor also at King Abdulaziz University of Science and Technology in Riyadh for several years. And also he was as a chair of the biotechnology at AGU, also as a visiting professor in the period between 2009 or 2007.
2007
to 2010.
2011.
So he has been very helpful and useful to our students and to the university. He has many years experience. I can say more than Halla. If Halla has 44 years experience, he has 45. She's not here, so I
My
can
God.
say.
My God.
I wanted to make you happy when I said I worked with flow cytometry before you were born, but that time when you were born, I was a baby. And also he published many papers in high-rated journals like Journal of Biological Science and Biochemistry, etc., in flow cytometry. And he helped also our PhD student who since our master. So you are heartily welcome. We are really happy to have you with us this week. I'm sorry for what had happened. I don't know whom to play in the plane, or the driver, or Huda. But not fun. So Professor Ehammen will talk about a balance of cell proliferation, cell death, and differentiation. And he will give you also more information about flow cytometry. And I hope it will be very useful. I apologize, I have to leave now to the hospital. And I will see you, inshallah, this week several times. Okay, thank you so much.
Thank you, Professor Moyes, for this introduction. Good morning, all my friends. It's true, all my friends. And I'm happy to be here. And it's a great honor for me after 5 years in this wonderful country, this wonderful facility, and I enjoy it. The first lecture, I will start with cancer biology, a very complex topic. So we will see how complex all these parameters, you will see it, which are. And the balance of cell proliferation, differentiation, and cell death is described by a regulation problem. And this regulation mechanisms of apoptosis and proliferation. The key points are growth can be altered by changing proliferation, apoptosis, or differentiation. Different cancers use different options to achieve uncontrolled growth. Proliferation is regulated by several checkpoints inside the cell cycle. And the G1 checkpoint is a focus for negative and or positive extracellular signals. In normal cells, the most of these factors are hypophosphorylated. For example, the RB, the retinoblastoma protein. Also, the P53 protein. It's also regulated by phosphorylation. The inhibition is relieved by cyclin-dependent serine-threonine protein kinases, the so-called CDKs. The CDKs are subject to stimulate the cyclin and inhibitors the CDKE. Controls mediated by protein-protein complexes, phosphorylations, and the cell cycle is regulated at different stages by altered activities of specific CDKs or CDKs, and also the cyclins. Alterations in the cyclins and the CDKs have been detected in cancers. And all these factors, it's possible to detect by flow cytometry. And two types of cell death, the so-called apoptosis or the programmed cell death, requires RNA and protein synthesis. Necrosis does not. Apoptosis is activated by DNA damage, withdrawal of growth stimulatory cytokines, or addition of cell death promoting cytokines. Diverse apoptosis signals converge to elder mitochondrial functions. There are the pro-apoptosis factors, for example, the BAX and the BAT, and anti-apoptotic proteins. Anti-apoptotic is a BCL2 and also the complete BCL family. And this regulates the release of mitochondrial cytochrome C. Apoptosis can be regulated by altering the relative proportions of pro-apoptotic and anti-apoptotic proteins. Cytochrome C activates protealysis of the caspase family. Proliferation and the apoptosis are integrated by mechanisms involving P53 and also the RB. Leukemias result from blocked differentiation, methylation of cytokine base in regulatory regions of the DNA contributes to differentiation. Growth is a general term indicating alteration in size of a cell mass and its end product of several interrelated influences such as proliferation, differentiation, cell death, cell contacts, and also the blood supply. If the time taken for cultured cells to double their number is compared with the time required for a tumor to double its volume in the patients, then there is a discrepancy. The cultures doubles in a few days. In patients, the doubling could be months or even years. This is a great difference between cell culture and patient material. The regulatory processes are present in solid cell masses but are absent in dispersed cell cultures. Cell culture conditions have been designed to maximize proliferation and to minimize negative influences such as the cell death. In patients, changes in cell number represents a balance between the proliferation and the cell death. So alterations of either parameters can influence the size of the cellular mass. Additionally, if cells differentiate, their proliferation potential decreases, and so differentiation has a negative influence of the cell growth. Thus, in normal tissue, cell number remains constant because the balance between proliferation, cell death, and differentiation is, you can see it. The number of proliferation cells remains constant. You see as the blue proliferating cells, the red one dead cells, and the green one differentiated cells. In abnormal situations, increased cell number can result either from a blocked cell death and or differentiation, or from increased proliferation, with no change in the other two properties. Each of these routes is involved in carcinogenesis. And this is the situation if you see a block, only proliferating cells are visible. No differentiation and also no cell death, or only few cell death cells. Increased proliferation is no change in cell death or differentiation. The number of proliferating cells increased, the blue one, only few are going to cell death, and only a few are cells which are differentiations. The normal proliferation and its regulation is starting with the cell cycle. For a cell to divide, the DNA must be replicated and distributed equally to their both daughter cells. This process of DNA synthesis and mitosis are separated by gaps. You have heard it's the G1 phase, the G2 phase, between the S phase, and also the mitosis, during which RNA and protein are made, and the cell reorganizes itself for the next round of the cell division. These series are called cell cycle. It's starting with a G1 phase and sometimes divide to include a G0 phase. But between G1 phase and G0, you cannot discriminate. It's not possible because the DNA content is the same. And this G0 phase, its meaning are quiescent cells. Differentiations take cells out of the cell cycle and can be represented as an exit from the G1 phase. Normal cells have a diploid DNA content. The DNA content doubles during the next phase of the cell cycle. Exit and join the G1 phase generate non-diploidic cells. Exit in the second gap, the G2 phase, between DNA synthesis and mitosis, produce tetraploid cells. Tetraploid cells means the double of chromosomes in a cell. During this complex series of events leading to the cancer formation, that means chromosomal DNA changes occur that generate DNA contents per cell that are simple multiplies of those of the diploid cells. And such cells are said to be polyploid or aneuploid. And the most tumors, breast cancer, tumomas, carcinomas, renal carcinomas, are mostly aneuploid. The DNA content in normal cells is 5.8 picogram. That means 5.8 10 up 12 gram is diploid DNA content. Tumor cells, the DNA content is mostly more than 5.8 picogram. Only few tumors shows a loss of 5.8 picogram, but only in 1%. And this with more than 5.8 picogram, it's polyploid or aneuploid. The G1 phase, it's the first step phase, is the most variable of the cell phases. Its length being the major determinant of cell cycle time, and it's also the period in which future commitment to division, differentiation, or cell death is made. And it's focal point for important signals. It follows that these signals are altered, then the cell cycle is affected. Many changes in gene products have been identified in G1, and it's convenient to classify these changes into early and late events. Examples for these early genes are the oncogene C-NYC and C-JON. Also, the primary regulation of the G1 checkpoint involves three families that are the cyclins, cyclin-dependent serine protein kinases, the so-called CDKs, and cyclin-dependent kinases, inhibitors, CDKs. One key substrate of the kinase is a protein of the retinoblastoma gene. It's, in short, it's the RB gene. This protein blocks the cell proliferation at the G1 checkpoint. And this suppression is released by protein phosphorylation and regained by dephosphorylation via protein phosphatases. The second element of the G1 regulation involves the DNA repair. DNA damage is detected by a mechanism involving the suppressor gene P53, which blocks the cell cycle progression until repair is completed. And in normal cells, P53 levels are very, very low. But its transcription is increased by DNA damage. For example, you have X-ray to a cell, then the P53 level increase very, very strongly. So, where are the P53 checkpoints? You see here the cell cycle, G1 phase, the S phase, G2 phase, the M, the mitosis. And you see the checkpoints are at the end of the G1 phase and also at the end of the synthesis. And the example for X-ray or chemicals leads to a DNA damage. And P53 is increasing, and this leads to a cell cycle arrest. So, in case a cell is able to repair the DNA, the cell cycle is running further. The next step is the cell cycle arrest, and you have irreparable damage of the DNA. And this leads to apoptosis and damage and potential cancerous cells. The S phase, the synthesis phase, is controlled larger at the level of its initiation and to build up of enzymes, regulatory proteins, nucleotide 3 phosphases. At the G1 S phase boundary provides that's the control. During the S phase, when the DNA strands are separated, bases are exposed and therefore are sensitive to external agents such as drugs or mutagents or X-ray. Given the fundamental importance of the DNA replication, defects in synthesis would be lethal. Sorry. Mitosis, the sister chromatids must be aligned correctly so that they are refracted to opposite poles of the dividing cell. This is monitored at the spindle assembly checkpoint. Limited data indicate that regulation is achieved at the point of attachment of microtubules to the centromere. This occurs through a chromosome structure known as a kinochore, which affects some microtubules. If any centromeres are not attached to the microtubules, then the mitosis is delayed. Special structures, sometimes in cancer cells, you see triplo centromeres. An additional regulation occurs if inappropriate attention of abnormal dynamics of the cell of the microtubule are detected. Once again, the CDKs are required for these events. If any of those events are wrong, such the cells traverse S phase but not the M phase. And this increases aneuploidy occur. And this is an example for the cyclins. You see on the x-axis, the x-axis, the G1 phase, the S phase, G2, and the M phase. Cyclin D is overall the cell cycle. Cyclin E is only present in the S phase. Cyclin A is only present in the G2 phase, and cyclin B in the M phase. That means in the mitosis. And you can measure this also by flow cytometry. It's complicated to measure this, but it's running. So cycling will increase, the cycling D especially, in breast cancer and esophagus cancers. And also in sarcomas, glioblastomas, and also in melanomas. A decreasing is visible in pancreas carcinomas and also in glioblastomas. And where but indirect effect is a P53 induced P21 in the breast cancer and colon cancer, and in few cases in eyes and lung. Proliferation of cancers are characterized by their unregulated proliferation. That means they have a lower requirement for growth factors, and they do not respond to negative environmental stimuli such as contact with other cells. These changes feed into the system mainly via the G1 checkpoint, and many of the characteristic variations used by different cancers occur in those pathways. These alterations of in cyclin and CDK genesis are important for the genesis of certain cancers. But two genes are most frequently altered in these cancers, which are P53 and the RB. You see here is the chromosome 13 for the ABs. Here is the localization, and also chromosome 17 and the localization for P53. And these two genes are achieved by mutations or deletions of the genes itself, or by binding of the normal product to other proteins. All of the synthetized levels changes in P53 are more dangerous because alterations of only one allele. Senescence and cell mortality. A major distinction between cancer cells and normal cells is that before senescence, cancer cells undergo more cell cycles of proliferation than normal cells. Cancer cells are said to be immortal. Also, this is not strictly true, as they do die also eventually. In cell cultures, you see a lot of cells die. Nobody knows. This prolonged life provides more time in which the accumulation of genetic errors which have attendant effects on cell function. Normal cells must therefore have a mechanism for limiting the life of a cell lineage that is lost in cancer. Cells are alive but arrested in the G1 phase of the cell cycle. There is not an absolute block of their transition into the S phase until the further results in apoptotic cell death. It originated when cultured cell lines were beginning produced from primary cultures. The primary culture eventually stopped proliferation. So during the beginning of the primary culture, mostly at the 3rd to 5th passages, they stopped proliferation. I have seen this in several primary cultures. But foci, the clones of cells, would spontaneously be reactivated and could be recultured as servile. You find these effects by cloning. A lot of your clones you pick, the most